Hello, everyone. I thought we would talk about more about the rain. I want to share a little video with you. It's 10 minutes long. How close are we to harnessing synthetic life? Hold on. Sorry about that. I want to make the full screen.
Like software. What if we told you that in the not too distant future, you could log onto your computer and program living cells like software? Ever since scientists unlocked the secret language of biology, DNA, we've made massive strides in understanding the base code that underlies all living things. And over the past few decades, we've become so obsessed with this code that scientists are now exploring how to edit genomes and even create brand new ones that never existed before. So, how close are we to harnessing synthetic life?
Life is a technology. It's a technology we didn't create.
But what if we could learn to harness it? That's the principle behind synthetic biology, the emerging study of building living systems. You might be imagining armies of Frankenstein creatures, and we'll get to that. But what we mean for the time being is creating animal products or individualized medical therapies, even transplantable organs, all starting with synthetic DNA and cells in a lab.
I look at them as 3D printers, but the most powerful and successful 3D printers that exist today, because they can print thousands and thousands of different materials all at once. And they can make more of themselves. And it comes with a programming language, DNA.
Pulling the principles of engineering design and then the real fundamentals of molecular biology together is really powerful, because it makes you think of biology as a design kind of medium, if you like, using the genetic material as the building blocks.
And when it comes to synthesizing life, there are two main schools of thought.
One is actually trying to build artificial cells. So actually starting from the bottom up and saying, how do I build a living cell in a kind of synthetic way? You either using sort of synthetic chemical components or using natural biological components. How do I build a cell from first principles, from scratch, basically, bottom up?
In 2010, Craig Venter's research team made history with this approach when they created the world's first synthetic organism, a bacterium made entirely of synthetic DNA.
And most recently, out of the UK, Jason Chin and his team synthesized the E. coli genome. That's pretty remarkable. That's a 4 million base pair genome.
So we now can specify we want a genetic sequence, A, T, G, C, T, 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 A, whatever. And we can send that information across the web to a company, and then they will put it into their production pipeline, and then we will get sent back a chemical synthesized piece of DNA.
It's only going to get faster and cheaper and easier from here.
But it's still not as fast and cheap as editing an existing genome to synthesize new life. And to be honest, starting from scratch might not be worth it.
A purist would say you have to make all the cellular components from atoms. Okay, no cheating, no using bits of life. You can make fully synthetic DNA chemically, but for most purposes, it's not any more useful than something that's semi-synthetic.
Now, the other way is actually taking existing cells but re-engineering them to have particular functionality, to the extent of completely resynthesizing their whole genome and redesigning it, but putting it back into the cell, which means it's still a species, but it's got a human-designed genome in it. Building that code to perform a function or to do a design or to make something.
And that's just what we're beginning to explore. With genetic engineering tools becoming more accessible than ever before, researchers want to use synthesized genomes to enhance human health.
For simple things like detecting infections or detecting environmental pollutants, we can now engineer bacterial cells that will detect arsenic or fluorine or any of these toxic chemicals, and will change color like a pH strip.
These bacteria could protect us from consuming toxins in contaminated drinking water, for example. And Andrew and his team at Humane Genomics are working to develop a library of synthetic viruses that could lead to customized cancer therapies.
So the virus is like a USB stick. You know it's going to dock with the particular cancer cell. And it's going to load a program into that cell. Now we get to write that program to make it even more specific for the cancer cell. We're working with dogs and dog cancers to gain experience. The cancers that we're working with, first, it's a bone cancer. So we put in little switches into the viral genome so it's only active in a bone cancer. And you can measure its killing activity relative to normal cells.
Sorry, I'll skip this in just a second.
Like the first attempt at anything, it's not going to cure cancer. But we demonstrated that we could go from a file on a computer to a virus particle that could infect a cancer cell. And we did it in a relatively short period of time for a relatively low cost.
Other researchers hope to take on the fashion industry with lab-made versions of materials like leather or silk. But perhaps the most ambitious plan involves bioremediation, preventing or counterbalancing ecological damage using microorganisms.
The concept is you have organisms that are engineered that would go out and either, start degrading plastic, start using plastic as an energy source, possibly start using toxic components, cleaning up environments using natural microbial systems.
Okay, we know what you're thinking. What about synthetic humans? Enter Human Genome Project Right, or just Genome Project Right. A sequel to the Human Genome Project, GP Right is bringing together over 100 institutions worldwide in one communal quest to synthesize the human genome.
You want a human cell which has new properties. You want to make something that has new properties like virus resistance or senescence, aging resistance. That you could do it with organic chemistry, or you could do it with biochemistry, or you could do it by editing of living cells. You could build human parts, meaning transplantable human parts. And from that, you could build large portions of a human being. But right now, I think we're in a time where that's on hold, in part for lack of strong arguments for why you would want to do it.
Instead, these researchers are looking to understand the genetic symphony that makes humans tick by demystifying our cells' instruction sets.
The simplest microbe in the world, right, which has got the smallest number of instruction sets, we don't even know what 40% of those instructions do. Our knowledge base is still quite limited. So it's like redesigning an airplane with components, and half of the components you don't know how they function. So that's where we're at.
And if you think tinkering with genomes is a bad idea when we know so little about them, you're not alone. I mean, what if something goes wrong? Should I live in fear of evil synthetic humanoids taking over the world?
I think fear and concern and dystopic narratives is a good thing. I think it's far better to have too much discussion of how things can go wrong than having too little.
We've been using living organisms to produce and manufacture things for thousands of years. We've been using yeast to make bread, alcohol, but what we're doing now is much more systematic and just opens up all the possibilities.
We have to frame it as the exploration of inner space. Instead of going up into the stars, we're going down into the cells. We're learning how to get there, how to explore, and how to manipulate. It's as important as going out to the stars, because if we get to Mars and we don't have a way to grow food and to keep ourselves healthy, we're not going to survive there for very long. We just have to start opening the doors and saying, come on in, take a look. Don't be afraid. Let's all learn and grow and use this incredible technology for good.
So, if DNA is really the next HTML, how close are we to harnessing synthetic life?
This is a very fast-moving field, and I think, in 10 years' time, 15 years' time, we will know enough about microbial genetic engineering to be building microbes that could be potentially quite novel.
We went from 2015 being able to edit 2 genes to 2017 doing 25 genes in the germline, and now we can do 26,000, which is actually relatively easy in the era of exponentially improving technology. I think we could have mammalian systems, including human, that are multivirus resistant within, I don't know, I'm guessing 2 to 6 years.
All you have to do is be able to write a new genome that nature hasn't created. And by that definition, I'd say we're already in the age of synthetic life.
We're quite far away from even thinking about a human, but as the technology increases, there'll be more interventions into multicellular organisms, and we need to be mindful of what they are and have good reasons for why we're doing it.
It's in your DNA to love science. So click the subscribe button and let us know down in the comments below or over on our...
Okay. I thought we would talk about some of this, but I'm going to now play this video from Craig Venter. Let's hear what he has to say about synthetic life.
I've talked about some of these projects before, about the human genome and what that might mean, and discovering new sets of genes. We're actually starting at a new point. We've been digitizing biology, and now we're trying to go from that digital code into a new phase of biology with designing and synthesizing life. So we've always been trying to ask big questions. What is life is something that I think many biologists have been trying to understand at various levels. We've tried various approaches, paring it down to minimal components. We've been digitizing it now for almost 20 years. When we sequenced the human genome, it was going from the analog world of biology into the digital world of the computer. Now we're trying to ask, can we regenerate life, or can we create new life out of this digital universe? This is the map of a small organism, Mycoplasma genitalium, that has the smallest genome for a species that can self-replicate in the laboratory. And we've been trying to just see if we can come up with an even smaller genome. We're able to knock out on the order of 100 genes out of the 500 or so that are here. But when we look at its metabolic map, it's relatively simple compared to ours. Trust me, this is simple. But when we look at all the genes that we can knock out one at a time, it's very unlikely that this would yield a living cell. So we decided the only way forward was to actually synthesize this chromosome so we could vary the components to ask some of these most fundamental questions. And so we started down the road of, can we synthesize a chromosome? Can chemistry permit making these really large molecules where we've never been before? And if we do, can we boot up a chromosome? A chromosome, by the way, is just a piece of inert chemical material. So our pace of digitizing life has been increasing at an exponential pace. Our ability to write the genetic code has been moving pretty slowly, but has been increasing. And our latest point would put it on now an exponential curve. We started this over 15 years ago. It took several stages, in fact, starting with a bioethical review before we did the first experiments. But it turns out synthesizing DNA is very difficult. There's tens of thousands of machines around the world that make small pieces of DNA, 30 to 50 letters in length. And it's a degenerate process, so the longer you make the piece, the more errors there are. So we had to create a new method for putting these little pieces together and correct all the errors. And this was our first attempt, starting with the digital information of the genome of 5X174. It's a small virus that kills bacteria. We designed the pieces, went through our error correction, and had a DNA molecule of about 5,000 letters. The exciting phase came when we took this piece of inert chemical and put it in a bacteria. And the bacteria started to read this genetic code, made the viral particles. The viral particles then were released from the cells and came back and killed the E. coli. I was talking to oil industry recently, and I said they clearly understood that model. They laugh more than you guys are. And so we think this is a situation where the software can actually build its own hardware in a biological system. But we wanted to go much larger. We wanted to build the entire bacterial chromosome. It's over 580,000 letters of genetic code. So we thought we'd build them in cassettes the size of the viruses, so we could actually vary the cassettes to understand what the actual components of a living cell are. Design is critical, and if you're starting with digital information in the computer, that digital information has to be really accurate. When we first sequenced this genome in 1995, the standard of accuracy was 1 error per 10,000 base pairs. We actually found on resequencing it 30 errors. Had we used that original sequence, it never would have been able to be booted up. Part of the design is designing pieces that are 50 letters long, that have to overlap with all the other 50 letter pieces to build smaller subunits. We have to design so they can go together. We design unique elements into this. You may have read that we put watermarks in. Think of this, we have a 4-letter genetic code, A, C, G, and T. Triplets of those letters code for roughly 20 amino acids, that there's a single letter designation for each of the amino acids. So we can use the genetic code to write out words, sentences, thoughts. Initially, all we did was autograph it. Some people were disappointed there was not poetry. We design these pieces so we can just chew back with enzymes. There's enzymes that repair them and put them together. And we started making pieces, starting with pieces that were 5,000 to 7,000 letters, put those together to make 24,000 letter pieces, then put sets of those going up to 72,000. At each stage, we grew up these pieces in abundance so we could sequence them, because we're trying to create a process that's extremely robust that you can see in a minute. We're trying to get to the point of automation. So this looks like a basketball playoff. When we get into these really large pieces, over 100,000 base pairs, they won't any longer grow readily in E. coli. It exhausts all the modern tools of molecular biology. And so we turned to other mechanisms. We knew there's a mechanism called homologous recombination that biology uses to repair DNA that can put pieces together. Here's an example of it. There's an organism called Deinococcus radiodurans that can take 3 million rads of radiation. You can see in the top panel, its chromosome just gets blown apart. 12 to 24 hours later, it put it back together exactly as it was before. We have thousands of organisms that can do this. These organisms can be totally desiccated. They can live in a vacuum. I am absolutely certain that life can exist in outer space, move around, find a new aqueous environment. In fact, NASA has shown a lot of this is out there. Here's an actual micrograph of the molecule we built using these processes, actually just using yeast mechanisms with the right design of the pieces. We put them in, yeast puts them together automatically. This is not an electron micrograph. This is just a regular photomicrograph. It's such a large molecule, we can see it with a light microscope. These are pictures over about a 6-second period. So this is the publication we had just a short while ago. This is over 580,000 letters of genetic code. It's the largest molecule ever made by humans of a defined structure. It's over 300 million molecular weight. If we print it out at a 10 font with no spacing, it takes 142 pages just to print this genetic code. Well, how do we boot up a chromosome? How do we activate this? Obviously, with a virus, it's pretty simple. It's much more complicated dealing with bacteria. It's also simpler when you go into eukaryotes like our cells. You can just pop out the nucleus and pop in another one, and that's what you've all heard about with cloning. With bacteria and archaea, the chromosome is integrated into the cell. But we recently showed that we can do a complete transplant of a chromosome from one cell to another and activate it. We purify the chromosome from one microbial species. Roughly, these two are as distant as human and mice. We had a few extra genes so we could select for this chromosome. We digested it with enzymes to kill all the proteins. And it was pretty stunning when we put this in the cell, and you'll appreciate our very sophisticated graphics here. The new chromosome went into the cell. In fact, we thought this might be as far as it went, but we tried to design the process a little bit further. This is a major mechanism of evolution right here. We find all kinds of species that have taken up a second chromosome or a third one from somewhere, adding thousands of new traits in a second to that species. So people who think of evolution as just one gene changing at a time have missed much of biology. There's enzymes called restriction enzymes that actually digest DNA. The chromosome that was in the cell doesn't have one. The chromosome we put in does. It got expressed and it recognized the other chromosome as foreign material, chewed it up. And so we ended up just with the cell with the new chromosome. It turned blue because of the genes we put in it. And with a very short period of time, all the characteristics of one species were lost, and it converted totally into the new species based on the new software that we put in the cell. All the proteins changed, the membranes changed. When we read the genetic code, it's exactly what we had transferred in. So this may sound like genomic alchemy, but we can, by moving the software of DNA around, change things quite dramatically. Now, I've argued this is not Genesis. This is building on 3.5 billion years of evolution. And I've argued that we're about to perhaps create a new version of the Cambrian explosion where there's massive new speciation based on this digital design. Why do this? I think this is pretty obvious in terms of some of the needs. We're about to go from 6.5 to 9 billion people over the next 40 years. To put it in context for myself, I was born in 1946. There's now 3 people on the planet for every one of us that existed in 1946. Within 40 years, there'll be 4. We have trouble feeding, providing fresh, clean water, medicines, fuel for the 6.5 billion. It's going to be a stretch to do it for 9. We use over 5 billion tons of coal, 30 billion plus barrels of oil. That's 100 million barrels a day. When we try to think of biological processes or any process to replace that, it's going to be a huge challenge. Then, of course, there's all that CO2 from this material that ends up in the atmosphere. We now, from our discovery around the world, have a database with about 20 million genes. And I like to think of these as the design components of the future. The electronics industry only had a dozen or so components, and look at the diversity that came out of that. We're limited here primarily by biological reality and our imagination. We now have techniques, because of these rapid methods of synthesis, to do what we're calling combinatorial genomics. We have the ability now to build a large robot that can make 1 million chromosomes a day. When you think of processing these 20 million different genes or trying to optimize processes to produce octane or to produce pharmaceuticals, new vaccines, we can change just with a small team, do more molecular biology than the last 20 years of all science. And it's just standard selection. We can select for viability, chemical or fuel production, vaccine production, etc. This is a screen snapshot of some true design software that we're working on to actually be able to sit down and design species in the computer. We don't know necessarily what way they look like. We know exactly what their genetic code looks like. We're focusing on now a 4th generation fuels. You've seen recently corn to ethanol is just a bad experiment. We have 2nd and 3rd generation fuels that will be coming out relatively soon that are sugar to much higher value fuels like octane or different types of butanol. But the only way we think that biology can have a major impact without further increasing the cost of food and limiting its availability is if we start with CO2 as its feedstock. And so we're working with designing cells to go down this road, and we think we'll have the first 4th generation fuels in about 18 months. Sunlight and CO2 is one method. But in our discovery around the world, we have all kinds of other methods. This is an organism we described in 1996. It lives in the deep ocean, about 1.5 miles deep, almost at boiling water temperatures. It takes CO2 to methane using molecular hydrogen as its energy source. We're looking to see if we can take captured CO2, which can easily be piped to sites, convert that CO2 back into fuel to drive this process. So in a short period of time, we think that we might be able to increase what the basic question is of what is life. We're truly, have modest goals of replacing the whole petrochemical industry. If you can't do that at TED, where can you? Becoming a major source of energy, but also we're now working on using these same tools to come up with instant sets of vaccines. You've seen this year with flu, we're always a year behind and $1 short when it comes to the right vaccine. I think that can be changed by building combinatorial vaccines in advance. Here's what the future may begin to look like with changing now the evolutionary tree, speeding up evolution with synthetic bacteria, archaea, and eventually eukaryotes. We're a ways away from improving people. Our goal is just to make sure that we have a chance to survive long enough to maybe do that. Thank you very much.
Okay. I will leave you with that. That's a lot to think about, lots to talk about. I'll be back soon with another episode of The Twilight Zone. Thanks for watching, guys.